Hello and welcome. My name is Xincheng Li from Jiangsu Normal University in China. Thank you for turning to this recorded presentation. The title of my article is Molecular Mechanisms of Antibiotic Resistance in Bacteria and Alternative Treatment Strategies from Evolution of Resistance to Precision Antimicrobial Therapy. Now, let's set the context. As you can see on the screen right now, I have highlighted the keywords in blue for you. According to the lesson predictions, by 2050, infections from resistant bacteria will cause 8.22 million deaths annually, 1.7 times the number in 2021. The World Health Organization has classified multi-drug resistant bacteria as a global public health threat requiring urgent international response measures. Confronting the problem of widespread resistant bacterial infections, which has been exacerbated by the pervasive overuse of the antibiotics. I have conducted a comprehensive review of the academic article to elucidate the mechanisms of resistance and the prevailing therapeutic approaches for multi-edible resistant bacteria. On the basis of my preliminary work, this research focused on addressing the following key questions. What are the primary molecular mechanisms of resistant bacteria? What existing alternative antimicrobial treatment strategies are available? How can precision antimicrobial therapy be achieved to improve treatment efficacy? A detailed exposition of the three above-mentioned points will be presented. As can be seen, the article has been divided into 4 parts. The first part, main molecular mechanisms of resistant bacteria. The second part, challenges and impacts caused by resistance. The third part, alternative treatment methods. The fourth part, conclusion and future development directions. Firstly, I want to introduce the main molecular mechanisms of resistant bacteria to you. Prior to elaborating on the resistant mechanisms, let's think about the basic question: how do drug-resistant mechanisms arise? And here are my considerations. Antibiotic resistance is a result of bacterial adaptation to environmental pressure, primarily acquired through spontaneous gene mutations and horizontal gene transfer. Spontaneous gene mutations can lead to bacteria acquiring resistance, with these mutations potentially affecting antibiotic targets, membrane permeability, or drug metabolisms. To summarize, the above constitutes the biological basis of antibiotic resistance. Now, let's turn to the next topic. As can be seen, the PPT briefly elaborates on the 4 main resistance mechanisms. For a detailed explanation of these mechanisms, please refer to my article. The next part of my article is the challenges and impacts caused by resistance. Among all the challenges, the most formidable one is the clinical challenge. With the spread of resistance, clinically viable antibiotics are becoming increasingly limited, and these drugs often come with serious adverse reactions. Resistant bacteria are the main pathogens in hospital-acquired infections. These infections not only prolong hospitalization and increase medical costs, but may also lead to serious complications or even death. Besides clinical challenges, the social and economic impact is also not to be overlooked. Resistant genes can spread in hospitals and communities through food chains, water sources, and international travel, posing a serious public health threat. The treatment costs of resistant bacteria infections is far higher than that of sensitive bacteria infections, including more expensive drugs, prolonged hospitalization, and additional isolation measure costs. Now let's turn to the next part. Existing treatment methods have significant limitations. Traditional antibiotics have limited therapeutic effects on multi-diable resistant and extensively drug-resistant chains. The development of new antibiotics faces numerous challenges, including long development cycles, low success rates, low return on investment, and technical bottlenecks. These factors collectively lead to insufficient antibiotic innovation, failing to meet the urgent need to combat resistant bacteria. In the following, I will introduce several common alternative treatment methods. The first strategy is phage therapy. Bacteriophages are viruses that specifically infect bacteria, completing their life cycle by lysing host bacteria. Phage therapy, as an alternative to antibiotics, offers advantages including high specificity, self-replication, biofilm penetration ability, and high safety. Through genetic engineering technology, scientists can modify bacteriophages to enhance their therapeutic effects, including expanding host range, enhancing lytic ability, improving stability, and delivering CRISPR-Cas systems. The second strategy is antimicrobial peptides. Antimicrobial peptides are short peptides produced by organisms with broad-spectrum antimicrobial activity, including those of animal, plant, and microbial origin. The mechanisms of action of antimicrobial peptides are diverse, mainly including membrane disruption, inhibition of intracellular targets, and immunomodulatory effects. The third strategy is gene editing technology. The CRISPR-Cas systems, as a revolutionary gene editing tool, can be used to target bacterial resistance genes, including specifically knocking out resistance genes, diversifying delivery systems, and developing antimicrobial CRISPR therapies. And the fourth strategy is nano-antimicrobial technology. Nanometal materials such as nanosilver, nanocopper possess unique antimicrobial properties, including direct contact killing, free radical production, mental ion release, and biofilm inhibition. Nanodrug delivery systems can improve the therapeutic effect of antibiotics, including targeted delivery, controlled release, enhanced permeability, and synergistic effects. Now, let's turn to the next strategies. Artificial intelligence-driven precision antimicrobial therapy. Artificial intelligence and machine learning technologies can be used in multiple aspects of resistant bacteria research. including resistance gene prediction, repeat resistance detection, resistance evolution modeling, and so on. Artificial intelligence has enormous potential in the development of novel antimicrobial drugs, including antimicrobial peptide design, drug target identification, drug repurposing, and combination therapy optimization. Now, let us summarize the content of the preceding text. This paper systematically summarizes the molecular mechanisms of resistant bacteria and alternative treatment strategies. The molecular mechanisms of resistant bacteria are diverse, including antibiotic inactivating enzyme production, target modification, efflux pump enhancement, reduced cell membrane permeability, and biofilm formation. To address the resistant crisis, various alternative treatment strategies have emerged, such as phage therapy, antimicrobial peptides, gene editing technology, and nano-antimicrobial materials. These novel methods exert antimicrobial efforts or effects through mechanisms different from traditional antibiotics, potentially breaking through resistant bottlenecks. Meanwhile, artificial intelligence and machine learning technologies provide new approaches for precision antimicrobial therapy, optimizing resistance detection and new drug development processes. Future research on resistant bacteria should focus on the following directions: in-depth analysis of resistance mechanisms, optimization of alternative treatment strategies, exploration of combination therapy regimens, improvement of clinical application feasibility, and development of precision medicine. Combating resistant bacteria requires global collaborative efforts, including strengthening global antimicrobial drug regulation, establishing resistance monitoring networks, promoting knowledge sharing and technology transfer, increasing research and development investment, and promoting interdisciplinary collaboration. Only through multifaceted, multi-level comprehensive responses can we effectively elevate the resistance crisis and safeguard global public health security. Thank you for your listening.